Thanks very much to Butch and Patrick and Ray, if you're out there. It's a real honor to be here. It's probably no other scientist that I admire more than Steve. You've all heard how lovable he is, and he's also a very bright guy, and I think he's better at a lot of the stuff that's done in quantitative genetics and PopGen is quite technical, as some of you might be aware of. And Steve has this gift of really distilling really complex problems down to terms that the rest of us are capable of understanding. I think the ticket for me to give a talk here I bought 31 years ago. This is a paper I wrote with Steve. I think we were locked up in an army barracks or something in Norway. And this meeting that was on size-structured populations, and a lot of fish stuff was going on at the time. And I was coming off my years as a limnologist, zooplankton ecologist, and we worry a lot about size-selective predation and things. And Steve and I got talking about this, and we realized that we talk about selection on size. Size is a consequence of growth over many intervals, and we never really are sure that it's actually not selection on growth rate at different intervals as opposed to the final size you're measuring. And so we use this path analysis. You may be able to see these should be called Arnold diagrams, I guess, by this point. But you'll see Z2 is like the size of an individual at point 2 in life, and you start with a Z0 and a Z1, and then there's these delta Z values. Those are the growth rates. So this would be a G matrix of growth rates and sizes, and the idea was to try and figure out how to measure selection on both growth rate and size as separate sort of traits. And then we realized that this is a problem because some individuals, because their size-selective predation, are dead before they express certain traits. So how do you reconstruct the whole cigar, or the whole G matrix, when certain traits you can't measure because the individuals have been censored? And we figure out, I would say Steve figured out a way to work backwards and get this G matrix. So this paper has been cited 37 times since 1988. It's about a little more than once a year, and this is not padding. Steve has never cited the paper. And I believe I've cited it once. And as far as I could tell, of all the people who did cite the paper, never really used the technique. But I still think it's quite a neat paper. Mark Kirkpatrick wrote a more elegant, developed some more elegant theory associated with growth trajectories and so on about the same time. Maybe that's what happened to us. So I've kind of drifted off into different areas since I used to be at Oregon, the other Oregon University, for a while. And I've kind of drifted off into genomics, and then I've been drifting out of there recently into cell biology. I'm trying to start this new center for mechanisms of evolution at Arizona State, where we're more focused on evolution at the cell level. I like this because we have a really well-established field of molecular evolution, genome evolution. There's this stuff called evo-devo, but we just sort of jumped right over the cell. And yet there's extraordinarily detailed information, sort of the natural history of what goes on inside cells, a great way to connect to the real nuts and bolts of evolution. These are a few of the organisms we work on here. These are all ciliated protozoans. That's not a kiwi fruit down there on the right. So I give a lot of talk to cell bio audiences, so I don't want to dumb this down too much, but cell biology is usually the last paragraph of every paper in Cell or any molecular journal. There's some statement, what is it, wondrous forms most, you just do wondrous forms most beautiful mumbo jumbo and say, I've measured this in 2 organisms and here's the selective force that caused these changes. Even the most arcane things that must be due to natural selection because that's the only evolutionary force. But we know what selection can and can't do depends on what we call the population genetic environment. And so this has got these 3 dimensions. Mutation, of course, creates variation upon which natural selection operates. Recombination reassorts variation. Then there's this mystical force called random genetic drift, which isn't so mystical to us, but I usually have to demystify it, and then the cell biologists have this holy cow moment. It's real. So most of you know that part of random genetic drift is just fluctuations in numbers of individual genotypes from or alleles from generation to generation. So I don't really need to go through that. What I do want to emphasize is it's not just numbers that determine the stochasticity in evolution. In fact, once numbers get really large, probably most of the stochasticity is due to this way we reconstruct our genome to have all our genes on these big, long strings of DNA called chromosomes, and that means that mutations that arise in individual genes can also be victims of all the other things in the surrounding neighborhood for moderately long periods of time before there's recombination. And as a consequence of that, you can get selective interference, whereas with free recombination, you'd fix two beneficial mutations. You sometimes can't do that when there's a selective sweep, and some of the other beneficial mutations get left behind. And you can even drag deleterious mutations along with another mutation that's more beneficial. So all these things add up to noise in the evolutionary process. And this is leads to a general concept that we've been trying to develop and extend to a bunch of cellular traits across the tree of life. I call this the drift barrier hypothesis, and the idea is pretty simple. So imagine you have some trait that's under relentless selection to increase, like enzyme catalytic efficiency or something, and then think about this trait being under the same selection in populations of different sizes. And you're relentlessly pushing towards this thing you could call molecular perfection. That's the biophysical limit. So say for enzyme catalytic efficiency, that would be that your enzyme's so perfect that the only thing limiting the reaction rate is the diffusion rate of the enzyme substrate. But as you move to the left there, random genetic drift is playing a more important role. And so it's sort of glazing the eyes of natural selection over, and we expect to be less perfect traits at the molecular and cellular level. So that's the basic idea. Now, one thing about this population genetic environment, that variation that I want to emphasize, is that all three of those dimensions vary by at least 3 to 4 orders of magnitude across the tree of life. And many aspects of evolution are ratios of individual components of the population genetic environment, like the ratio of the mutation rate to NE. So what happens in different phylogenetic lineages is really quite dramatic. Am I going backwards, or did you just see this? Yeah, okay. Okay, so one of the things that we're able to do now is to make some statement about this very difficult number to measure, the genetic effective population size. And we get this from polymorphism data in natural populations and known mutation rates. And I won't go through the details. I'm sort of skipping over the theory. I just want to get the main point across here. And this is a plot of genetic effective population sizes this is organisms across the tree of life. There's no archaebacteria in here, but otherwise we've got fairly broad coverage here. There's about 20 orders of magnitude of adult size in individuals here. And these are log-log plots here, and you can see that there's this sort of inverse relationship. Now, we big vertebrates live down in the lower right-hand corner, and typically the effective size is going to be on the order of 10 to the 4th individuals or so. What that means is that there's beneficial mutations with effects smaller than 10 to the -4, the natural selection just can't see them, and they're not available for utilization by selection. And in contrast, any deleterious mutation with effects smaller than that can't be seen by natural selection either and can't be eradicated. Now I move to the upper left, and all those black dots up there, those are bacterial species. And what's quite interesting here is that even bacterial species seem never to have an effective population size much greater than about 1 billion individuals. So all populations are finite, and probably mostly due to this genetic interference and that goes on with chromosomes. This keeps stochasticity still operating at a fairly high level with microbial populations, but still you can see that if you've got an effective size of 10 to the 8th, you can take advantage of mutations with effects as small as 10 to the -8. So there's this whole large window and granularity of mutational effects that microbes can take advantage of that we cannot. And this is important because there's been a lot of work over recent years trying to get a rough estimate of the distribution of mutational effects. And there's a lot of uncertainties about this, but I think we can say fairly safely that a large fraction of mutations at the molecular level are down in the effectively neutral range, especially for us big, clunky, multicellular things. And so those are the mutations that are in some lineages are open to and others closed to evolutionary exploitation. To give you a more practical view of this, think of just an insertion of a single base pair in a genome of something that has no function whatsoever. That's going to cost you about 100 ATPs. We know this from basic biochemistry. We also know that it costs about 10 to the 10th ATPs just to build a bacterial-sized cell. So the penalty you pay for just adding one base pair to a E. coli-sized genome is about 10 to the minus 8. You can think about that as a selective disadvantage just due to bioenergetics alone for inserting just one base pair in bacteria. And bacteria can feel this. And this is probably the explanation for why they have such streamlined genomes. But for somebody like us, you can insert like a whole mitochondrion on one of our nuclear genomes. And our cells have a much bigger total energy budget, so we would not even be able to feel this in terms of the effect this has relative to the power of drift. So we've been trying to exploit this drift barrier hypothesis in a number of ways. I've worked on this in the area of genome evolution and comparative genome architecture in different bugs. More recently, we've been working on mutation rate evolution, and I'll talk about that in a little bit of detail. But this is really a prelude to something I've been working on more recently, and I'm really happy that I realized this just before I came here. I didn't know what I was going to talk about that would relate to something I'd done previously with Steve. And it turns out we're now doing some work on growth rate evolution across the tree of life. So I think this will bring us back to the beginning of the talk. I hope so anyhow. It's sort of unfinished work. One way to think about the drift barrier hypothesis with respect to mutation rate, and again, I'm not presenting the general theory, but going way back to Kimura and others, including myself, there's quite a bit of theory worked out on how natural selection operates on the mutation rate. Most mutations are deleterious, so we think in general selection will drive down the mutation rate. It's because if you're a mutator allele, you end up being linked to this deleterious load you produce. And things are even weaker in terms of selection if you're sexual because you become disconnected from the mutation load you are creating. But the main point I want to make is we think that natural selection is generally driving down the mutation rate. And as a consequence of this, you could have different starting points. So in the upper left, what we're looking at is the average mutation rate over time. And the scale on the lower right is just to indicate that there's alleles, many alleles at many loci in all organisms that influence the mutation rate. So from one sort of DNA polymerase, you can get a mutator allele or an anti-mutator. And it's not hard to do this in the lab. So there's a lot of genetic variation for the mutation rate. And so on the upper left, you see we start way up with a very bad situation, a very high mutation rate, and eventually you wander down in this gray area, and that's the drift barrier zone. And the lower blue line there, we're starting with a really good situation. You've got really high replication fidelity, but the selective advantage of it is low enough that natural selection can't keep you there, and you wander back up into the gray zone. And then you just wander around that zone forevermore. If you increase the population size effective population size by 100, the same thing happens, but the drift barrier is changed by a factor of 100. And this leads to the prediction there'll be this inverse relationship between the evolved mutation rate and the genetic effective population size of an organism. So we've been doing these things called mutation accumulation experiments for a long time. We started these at Oregon, University of Oregon when I was here, and they're kind of mind-bogglingly simple experiments to do, but they do take a lot of time, and I guess you eliminate a lot of technicians over the years and so on out of complete boredom. But in the end, you get a lot of good information out of these. So what we do is we start with a single genotype. Often this will be an asexual or self line, and then we replicate into 50 lines or so. And then we take these through a single individual bottleneck for often up to 3,000 generations or so. And what's happening here is that all the lines are isolated. You've maximized random genetic drift, so any mutation that's not lethal or causes sterility gets accumulated in these lines. The lines always get sick, and that's just consistent with the idea that most mutations are deleterious. And nowadays, we're mostly focused on the molecular end of things. So at the end of the experiment, we grind the individuals up, we isolate the DNA, we sequence the genomes to about 100x coverage, and then each line picks up its whole independent set of mutations. And we get not only the mutation rate, but the complete molecular spectrum. I'm not going to talk about the molecular spectrum today, but we've done this with quite a menagerie of different organisms. We don't do vertebrates. There's a lot of nice work now being done on vertebrates with trios. The vertebrate mutation rate's generally high enough, so you just take mom and dad and a few kids, and you got a good estimate of the mutation right now. But these are some of the bugs we've worked on, some of the eukaryotic bugs. We've even done an ichthyosporian here. Anybody in the room know what an ichthyosporian is? I do have $10. Joe? No? Okay. No one ever knows what an ichthyosporian is. I'll explain that one later, but it's a nice lineage below the metazoans. We've done about 30 species of bacteria. here. We're just about shut down with this kind of work. This is a picture. Yeah. Somebody knows a ichthyosporian?
Slime mold.
Slime mold? No, so we've done a slime mold. That's dictyostelium. Okay, you owe me $10. Okay. Yeah, this is not the one that kills people. It does kill, I shouldn't say this, it kills lizards. Yeah, it's a bad room to okay, so this is my current lab. You can see they're all smiling. This was right after I informed them we're never doing any more mutation accumulation experiments. And we've had pretty good funding to do this kind of work, as you might expect. It's conceptually simple, but it does cost some money to sequence all these genomes. At the end of the experiment, you just line up all the genomes. They all started out the same. This is a wheel diagram of 50 concentric circles, each a different genome of Bacillus, the soil bacterium. This is a thing called Mesoplasma florum. This is the smallest genome we can grow on a plate. It was isolated off the petal of a flower of a lemon tree in Florida. That's all I know about its biology, except we now know its mutation rate, which is 10 to the minus 8 is compared to 10 to the minus 10 for Bacillus. So that little bacterium has the same mutation rate as humans do per human generation. We're about 10 to the minus 8 per base. And this has got a generation of about 20 minutes. So I'm not going to go through all the details, but I just want to get to the drift barrier hypothesis and some data so we can get to the growth rate stuff. And so when you sum all these data up, we can plot here this is the base substitution mutation rate per generation as a function of the effective population size in these organisms. And you can see they're running downhill consistent with the theory. You can also see three different groups here. So the red are the multicellular species. And the blue and the green sets of data here are sort of overlapping in terms of effective size. The blue ones are bacteria. The green ones are unicellular eukaryotes. And you can see that what we've learned is that in general, for the same effective population size, unicellular eukaryotes are actually better at replicating and repairing DNA than bacteria are. And this makes complete sense in the context of the drift barrier hypothesis because selection is not really operating so much on the number of mutations per base. It's on the total number of deleterious mutations per genome. And eukaryotes have substantially larger genomes than bacteria do. So now if you take this x-axis and multiply it by the number of mutations, number of sites, nucleotide sites under selection, then all the data are more or less unified here. Those dashed lines, that's just a guide to a slope of -1. So that's sort of what we roughly predict by the drift barrier hypothesis. Those black arrows there are meant to suggest that some of these points should be elevated a bit because the number of nucleotides under selection here is just assumed to be the proteome size, but in us big, and that's essentially true for bacteria. That's about all there is to a bacterial genome. But for multicellular eukaryotes, there can be substantial amount of DNA associated with regulatory regions and so on. And that will only tip the plot up to make it a little more consistent with a slope of -1. So this is the first, to my knowledge, the first general theory for the evolution of a trait across the entire tree of life. And we're quite open to, my days are numbered, so we're quite open to alternative suggestions. I don't want to waste anybody's time on this, but this is what we think provides a general explanation for the tremendous variation, 3 to 4 orders of magnitude variation of mutation rates across the tree of life. Now, when you think about it, there aren't a whole lot of traits that you can measure across the tree of life, but everybody does have to replicate their genome. So that's one. Another one that I now want to finish up talking about is growth rate. All of us have to grow a bit. Even unicellular organisms have to double their cell size before they divide in general. And so one question is, can we extend these general ideas to other traits that we can measure in a very, very wide sense? Many of you have seen these kinds of scale. I really wanted to make a slide, a tic-tac-toe game up here, if anybody can do this before we finish the meeting. Couldn't quite synchronize the axes with the things up there, but many of you have seen these things that ecologists call power law behaviors, these scaling distributions of various traits, usually against organism size, and they're usually log-log plots and roughly a straight line, and so that's a straight line on a log-log plot, so that's why they're called scaling laws. The one on the left, this happens to be for population rate of growth of different organisms, different size. And again, you can see there's 15 to 20 or so orders of magnitude. So these are general patterns that one sees across the tree of life. The one on the right, this is basal metabolic rate scaled to the weight in different groups of mammals. And there's been lots and lots of sometimes acrimonious debates about what drives these scaling laws. And some people think they're fractal properties of organisms. There's just surface to volume ratio arguments. And in one camp, the prediction is a minus 1/3 scaling. In another camp, it's minus 1/4. And lo and behold, usually the slope is about minus 0.27 or something. So this keeps the debate raging on forever. And you can see there's occasionally some nonlinearities and so on, but always what's underlying this debate is these things must be due to structural constraints in organisms. So the only debate is what is the structural constraint? And I'd like to entertain the idea that it's not just a structural constraint. This may be an evolutionary constraint related to the drift barrier hypothesis. Because remember, as we go across this axis of body size here, we're changing the effective population by about 4 orders of magnitude. So there's really mildly deleterious mutations that can accumulate in genomes of us big clunky things as opposed to microbes. So this is a different kind of scaling relationship here. I want to make clear this is not the same kind of thing that I just showed. This is a scaling with respect to size at maturity of different organisms. The trait on the y-axis is the maximum growth rate in terms of biomass in a species. So by what I mean by that is if you were looking at sort of a metazoan and you knew the growth rate of every day in its life history, the maximum is the life stage in which the growth rate is the highest. So we're just trying to figure out of this organism with this genome, what's the maximum possible rate you could make biomass if you stuck the organism in that life stage. And of course, for unicellular things, it's just the cellular growth rate. And this is, again, one of these scaling laws that seems to apply across the tree of life. And I want to emphasize that in many of these, there's about an order of magnitude range of variation around these power law relationships. A lot of that is just due to sampling noise and data being gathered by various means in different labs and so on. In any event, you can see there's this general negative relationship. So bigger things just can't make biomass as fast as smaller things. And so usually these rates for metazoans, these are in the earliest few cell stages in the organisms we're looking at. The adults are always growing more rapidly. So this is just, if you wanted to start a biotech company, the way to think about it is what life stage of what organism would you use if all you wanted to make was biomass? And it would be over to the left there at the peak if you're using a eukarya, it would be unicellular eukarya. Interestingly, for bacteria, the larger bacteria get, the higher the growth rate is. So bacteria actually get better as they get larger. We don't know. That will look pathetic. I know for those of you who know something about statistics, but there's not a whole lot of range of variation in bacteria. And if you spread things out, you can see this. And we're not the first ones to point this positive scaling in bacteria. So the idea here with respect to the drift barrier is we're moving from left to right. We're going from effective population sizes of about 10 to the 8th to about 10 to the 5 or so. And you can see these patterns even within groups. So all the black dots are crustaceans. So to the left, all those black dots, those would be microcrustaceans living in the zooplankton. And down to the lower right-hand corner, these would be things like lobsters and so on. There's a big range of variation for mollusks too, from like zebra mussel up to octopus and so on. So you can see these scalings both within and between groups. So the idea here is the drift barrier hypothesis, and this is bringing, I think, closing the loop back to the work I did early with Steve, although I don't really know how this is also ties to some of the other talks on micro versus macro evolution, because this is macro evolutionary pattern, not micro evolutionary pattern, but it is a relationship you can see between growth rate and the size of an organism. So the plausible hypothesis, I think, is that this reduction in growth rate or ability to accumulate biomass as you move to the right is simply because natural selection cannot take advantage or cannot eliminate a certain mildly deleterious mutation. So as we move to the right, our genomes are getting more and more bloated with excess DNA. And that we pay a price for that just in terms of bioenergetics. So that's the general idea here. I think it's consistent with the data we have from evolutionary genetics. It's consistent with the information we have from physiological ecology. And this is just a little bit of theory that tells us, let's suppose you have mutations with different effects, say 10 to the minus 4 in orange, 10 to the minus 5. These are beneficial mutations, or even 10 to the minus 6. And just say there's a beneficial and a deleterious allele, you can ask what would be the expected frequency of that allele as you move to the right in populations of larger and larger effective population sizes. And as you move to the right, selection rules, and you essentially go to fixation for the plus allele. Here, there's a mutation bias here, so there's, I think the way I wrote this down was the rate of mutation to deleterious mutations is 100 times higher than that of beneficial. So as you move to the left, selection's completely ineffective, and the equilibrium just is the mutation balance. And then you have this gradient. So those solid curves were developed, it comes from theory developed by Kimura, Mariam, and Crow a long time ago for freely recombining loci. And the dotted lines, those are lower. That would be what you would get if you had completely linked loci and you have all this selective interference going on at the same time. Now in reality, in real organisms, of course, there's a whole distribution of mutations at different effects. So what would be going on is some mixture of all these things together. But the most important thing is you move to the right, it's just making the point again that selection is getting more and more efficient at promoting even very, very mildly beneficial alleles. So I think this is consistent with everything coming out in molecular evolution, as well as I said with the other data. Now the big question here is, okay, we're just arm waving about this evolutionary theory, and those other guys are just arguing about all these physical explanations for these power scaling laws. How can we separate out the two? It's quite possible both are operating at the same time. And it's hard to figure out how to tease these out, but one way you might be able to do it is to do an evolutionary experiment. And the prediction would be if you grew things under lower effective population sizes, the evolved growth rate would eventually be lower. And I was talking to my lab about this a few weeks ago, and they reminded me, well, we've been doing an experiment for 5 years with E. coli. We have about 400 lines of E. coli we've been growing under various conditions. This is sort of a Lenski on steroids sort of situation. And one of the things we've been doing with E. coli for 5 years is growing different lines at dramatically different population sizes. And it turned out that one of my postdocs had measured the growth rates in lots of individuals from these lines. We sequence these lines about every 3 months or so. So we know a lot about the mutations that have accumulated. And this is the final research slide. And what you can see, so as you move to the right, that's that's a higher dilution factor. That actually means that more cells are being transmitted from day to day. I think that's about 1 billion cells in the populations that are being transmitted. And then to the far left, that's only 1,000 E. coli cells being transmitted to generation. So again, the guys on the left can only feel a narrow window of mutations because drift is so overwhelming. And what you can see is that in fact the evolved mutation rate in just 5 years in E. coli experiments, the MMR, MMR's mismatch repair, so these are souped up so they mutated about 100 times faster than the wild type. Either way, you can see that these lines going through, being maintained at larger population sizes are evolving elevated growth rates. So we think this is consistent with the hypothesis. And I wanted to end with this slide here. Someone sent me this the other day. I think it was the president of our university. Maybe he was trying to tell me something. And I looked at this, and then this is a great quote from Martin Luther King, who was on I guess he was on the board of trustees at Morehouse College. And I was just looking at this, and I think this is a really great description of Steve Arnold. Not just, I don't want to you might skip over, I don't think any of us, hopefully, are menaces to society, but the point here is that intelligence is not enough, and Steve has got intelligence, but character is important, and Steve has spades in character, and this is an unusual combination to find, so it really is just fantastic to be here today celebrating Steve. So thank you very much.